Hello. Yes, am I audible to the end? Okay. Okay. So, good morning to one and all present here. I extend my warm welcome to all the participants, attendees, guests, students, scientists, staffs, and colleagues to witness the event organized as part of year-long celebration of the 75th year of establishment of CSIR-CDRI. The Innovation in Healthcare series oration is a well-thought initiative by our director, Dr. Radha Rangarajan, to bring in researcher with exceptional scientific achievements to catalyze the process of ideation and innovation. We have already witnessed the exciting talks in the series by Dr. Debarka Sengupta, Dr. Bushra Atik, Dr. Samrat Mukhopadhyay, and Dr. Debabrata Maiti last year. Today's talk will be the first in this year's Innovation in Healthcare series oration. We are honored to have with us esteemed speaker, Professor Arvind Kumar Bansal from NIPER-Mohali, our chief guest, Dr. Prabodh Trivedi, Director CSIR-CIMAP. May I now invite the dignitaries to the dais, Professor Arvind Kumar Bansal, Dr. Prabodh Trivedi, Dr. Radha Rangarajan, Director CSIR-CDRI, and Dr. Prem Prakash Yadav on the dais, please. So may I now request Director Ma'am to welcome Professor Dr. Arvind Bansal by presenting a flower bouquet, please. Now I request Director Ma'am to welcome Dr. Prabodh Trivedi by presenting a bouquet of flowers, please. Okay. Thank you, ma'am. Now I request Director Ma'am for her welcome address, please.
Distinguished guests, faculty, students, guests joining us online, our special invitees, students from Lucknow University and Rameshwaram Institute of Technology and Management, and everyone else present here, warm welcome to you. We're delighted to have you with us here at this event under the Innovation in Healthcare series, as Jia just explained. This series is held once a quarter and hosts distinguished scientists whose research has a strong translational focus. This is the second year for this series. We skipped the first quarter because we had CTDDR and it was a very full calendar for the institution. So this is the fifth talk, and we're deeply honored that Professor Arvind Bansal accepted our invitation to be here today. Equally, we are honored that Dr. Prabodh Trivedi, Director CIMAP, is with us as chief guest. Dr. Prabodh Trivedi is, of course, no stranger to CDRI. As director of a sister laboratory, he has been a frequent visitor, and he was on our management council. He's been a collaborator with CDRI scientists and has been on monitoring committees for phytopharmaceutical product development. He is an expert in the field of plant biotechnology, particularly in pathway engineering for secondary metabolites. Thank you for joining us, Dr. Prabodh. What makes today's event very special that it is part of our year-long celebration of CDRI at 75. Established in 1951, CDRI has entered a milestone year, and such occasions give us opportunity to reflect on the past, successes and failures, chart a course for the future, and most importantly, renew our commitments. Our commitment is to develop drugs, diagnostics, and process technologies, engage in high-quality basic research, teach and train students, and ensure that our IP moves to industry to enable affordable healthcare. All this is possible only if we are well connected across the research landscape, which includes academics, clinicians, and industry practitioners. So, one of our major goals during this 75th year is to have as many open events as possible. So, as I mentioned earlier, we have students from Lucknow area academic institutions, but we're also live streaming for a virtual audience. So, we hope to expand our circle of collaborators and well-wishers through such outreach. Today, we have an exciting talk lined up by Professor Bansal. He's a well-known expert in the field of formulation development. He specializes in the material science of pharmaceutical substances, particularly small molecule therapeutics, and his talk today is going to give us insights into addressing formulation challenges during drug discovery and development. Drug formulation is a very, very important part of drug development. It is the process through which a pharmaceutically active compound is transformed into a form that is usable for the patient. So proper formulation development ensures that manufactured drugs are safe and accessible and that the effects of the drug remain predictable and reliable. It is also an exciting source of innovation, allowing pharmaceutical companies to tap into completely new markets and for patients to experience better efficacy and safety. So we really look forward to today's talk. And before I hand the stage back to Dr. Gayan, I'd like to thank the committee members who have made this event possible. Dr. Prem Prakash Yadav, who chairs the committee, Dr. Gayan, Dr. Neeti Kumar, and Dr. Arvind Chatri. We're deeply grateful for your time and effort and for really taking care of all of the details that were necessary for organizing today's event. And thank you very much to all of you in the audience for being here and for those online who are joining us. And I now request Gia to come back to the stage. Thank you.
Thank you, ma'am. So, a famous quote says, the best way to have a good idea is to have lots of ideas. It gives me immense pleasure to introduce the speaker of the first Innovation in Healthcare series for this year 2025, Professor Arvind Kumar Bansal from NIPER, that is National Institute of Pharmaceutical Education and Research, Mohali. Professor Bansal did his B.Pharm and M.Pharm from University of Delhi and then PhD in the field of pharmacy, specialization in pharmaceutics and drug delivery system from University of Delhi. And he joined NIPER-Mohali in 2000 and now he is working as professor and he also served as the head of the division of pharmaceutics in NIPER-Mohali. He also served as Dean of NIPER-Mohali during 21 to 24. He is principal investigator of Center of Excellence in Innovative and Sustainable Pharmaceutical Development, a joint venture between Granules India and NIPER-Mohali. Professor Bansal's outstanding research outputs include more than 200 publications, more than 13,000 citations, H-index of 60, 12 granted patents, more than 150 master students graduated from his lab, and 27 PhD students graduated under his guidance. His lab developed a novel platform technology called NanoCrisp, that is nanocrystalline solid dispersions, a bottom-up technique for the generation of nanocrystals. This technology is currently being scaled up for clinical evaluation with industry collaborator. His contributions are recognized globally. He has been awarded with several prestigious awards and he is on editorial boards of various peer-reviewed journals. He has been appointed as Technical Committee Member, Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, Government of India. In 2016, he was awarded the prestigious Fellow of American Association of Pharmaceutical Scientists, that is AAPS. And he also honored with Distinguished Educator and Researcher Award from American Association of Indian Pharmaceutical Scientists. He is recipient of OPPI Scientist Award. He is expert member of CDESCO and also member of FICCI, that is Federation of Indian Chamber of Commerce and Industry, in 2005. Professor Bansal has vast research experience in the field of pre-formulation profiling, formulation development, and nanotechnology in drug delivery systems, polymorphism, particle engineering, selection of salt forms, involvement of oral viability and improvement, QBD, functionality of excipients, solubility enhancement techniques, etc. So we are privileged to have Professor Bansal with us today to share his insight on the topic tackling solubility challenges during drug discovery and development. Please join me in welcoming him. Professor Bansal, please. Okay.
Very good morning to all of you. Director CSIR-CDRI, Dr. Rangarajan, Dr. Trivedi, senior scientists of the institute, students. I'm really very happy to be here today. This is a visit to an institution which played a very significant role not only in its own progress but progressing the idea of NIPER when it was incepted. Dr. Nityanand, who was the one of the directors of this institute, was a pivotal figure in creation of NIPER-Mohali. So thank you for the invitation, and I hope that some of the things that I discuss today, which have been ongoing research program in our lab, would be helpful to this diverse audience, as I understand that the formulation team is relatively small in CDRI, but more of other specializations are there. Before I start going through the slide deck, I would like to share some interesting facts. Post PhD, I started my career with the pharmaceutical industry as a research scientist and worked with two companies, JK Pharmaceuticals and Ranbaxy Research Labs. Ranbaxy, as you know, was a pioneer in many ways, not only in terms of taking the Indian generic products to the advanced markets, but they were the ones who also started the new drug discovery program in a very early stage. In a private corporate, there was obviously research going on CDRI and other public funded institutions. And during that period, I was lucky to be around and being only few of the PhD at that time used to be a rare event, especially in pharmaceutical sciences. I was chosen to support the pre-formulation team of the new drug discovery and research. And actually it did not happen by structure, it happened by a very interesting incident that happened. There was a molecule in the drug pipeline which gave excellent results in the in vitro assays, and the entire team, especially the chemists and the biologists, were very excited about the molecule. And then the next stage is to find out the absolute bioavailability of that compound in animal models, and when they started doing it, they were not able to dissolve it in anything. And that was the time when the formulation team was called or summoned by the new drug discovery team that we now have a problem that you need to solve. So we were handed over that compound. We started working on creating the intravenous formulation, and we found out that it does not dissolve in anything. It was virtually a rock, but since the molecule was exciting from the in vitro studies, we kept on making efforts and created a formulation which in our language is called as a cocktail formulation, by mixing a number of solvents together and dissolving it forcibly. Once that was given to the animals, all of them died because of the toxicity of the solvents. And then it was decided that why don't you start working on a oral formulation? Let us try the oral exposure. So same story, in the solid form, it did not give any exposure. So we tried out solution formulations because they are acceptable in the early stages of the animal studies, and again created a cocktail. Now the same molecule which was giving very exciting results in the in vitro assays did not again give any results after the oral administration. And then something very interesting happened. The entire team passed on the blame to the formulation team that the molecule is very good, but you are not able to formulate it properly. And then we thought of a very innovative idea of administering the solution to the animals, waiting for some time, sacrificing the animals, opening up the gastrointestinal tract, and to everyone's surprises, we saw crystals of the drug sitting in the GIT, although we had given a solution to start with. So what was happening that as soon as it was coming in contact with the predominantly aqueous environment, it was precipitating out. And we gave our verdict that this particular molecule cannot become a drug. And that was the first time Ranbaxy realized that the formulation development has some role to play in drug discovery right from the beginning. And our company decided to create a dedicated group for pre-formulation of the molecules. So there was an ascendancy in the status of the formulation development scientists. They were not just looked at creating generic products. We saw their role in the drug discovery program as well. So my experience and my learnings have been strengthened over the period of years, not only just by our own experience, but the way the industry has progressed. Earlier, the formulation scientists only used to be summoned when there was a problem in solubility or in delivery. Now, the pharmaceutical development scientist plays a very significant role right from the beginning in the drug discovery program. And as we go through the slides, you will see that one of the reasons why most of us get jobs is because of poor solubility. So the chemist actually doesn't like poor solubility, but we love it because it creates a lot of jobs for the pharmaceutical scientists. So to start with the talk, the title is Tackling Solubility Challenges During Drug Discovery and Development. I've tried to restrict it to the relevance for the institute, but even in the development of the generic product for which there is already a reference product, I've seen that this particular property plays a very huge role even in the development of the generic formulations. So that's the flow of the presentation. I'll not spend too much time in a classroom right of teaching talking of fundamental of solubilities, but focus more on the technologies that are available. And these are with a focus on whether they are translatable to the commercialization stage or not. There might be a huge number of technologies, but there are some which have proved successful not only in discovery settings, not only in development settings, but also at the commercial scale. So that's the problem statement. The developability of a molecule sometimes is thought only to come from potency and selectivity, but this has expanded now to the role of ADME and material properties. And in the material properties are properties like solubility, stability, permeability, hygroscopicity, what typically a formulation development scientist would focus on. Unless there is an overlap of all these properties, which is called as a sweet spot, you will not be able to get a molecule that can be developed into a drug. The important difference that has also come into existence is that rather than looking at the material properties later in the development, they are now being looked right from day one in the discovery setup. This is a well-known slide that the solubility as a problem has been increasing over the years, and if you look at the marketed drugs, almost half of them are poorly water soluble if you look at BCS class 2 and class 4 drugs. But if you look at the drugs that are in the pipeline, which means they are coming up for the clinical development, almost 90% of them have become poorly water soluble. That's partially to do the way we do the drug discovery process, but the fact remains that solubility has become a problem that cannot now be neglected. All of you must have heard about a very famous Lipinski's rule of fives, which was proposed by a Pfizer scientist, Christopher A. Lipinski, I think in 2010. And based on that, it has become a guiding principle as an exclusion criteria for molecules that are not good for development, especially from the oral route and in the domain of small molecules. And it is applicable only to the passively absorbed drug. If there is an active transport, then it does not have a rule. The same gentleman, he's still around, and he has written few very interesting articles, and there is a punchline that he gave in one of his articles that I like very much, and it says that solubility sounds incredibly low tech. It's not glamorous, but it is incredibly important if you want to be a winner in the area of drug discovery. So that's a statement which holds true even more in these days, that we tend to ignore the contribution of solubility in the overall development and pay the prices by failures late in the stage. In the field of drug discovery, you know, there is a statement, fail early, fail cheaply. So if something has to fail, let it fail as quickly as possible. We should not spend resources to a later stage before the failure comes. Now, in the different stages of drug discovery and development, this is the role solubility plays. So in fact, starting from the lead identification to lead optimization to preclinical development, everywhere solubility has a role. And it's very important, in fact, at the lead identification stage that you identify the solubility problem, because you then have the possibility of using chemistry-based interventions which become unavailable later on. So you can create a prodrug, you can create a drug analog, you can create a new salt form. So all those interventions must be done right at the stage before you identify it as a lead molecule. Because if you do it later on, then you have to repeat all these studies, which the pharmaceutical company is never going to like. And that is why they emphasize that it has to be done early on. And look at this particular aspect. This is the animal studies. The optimal formulation should be locked at this stage. Formulation that has to go into commercialization should be locked right at the stage of preclinical studies. So there has to be a clear line of strategy that if you are doing preclinical studies with a particular formulation, it must be commercializable and scalable to the level that it becomes your marketed product. Obviously, if at all your product reaches to that stage. Any changes later on will call for lot of iterations, lot of equivalency studies, lot of bridging studies, which not only is time consuming, resource consuming, but can also give you some failures. Similarly, in clinical phase 1, phase 2 and through, and even after the regulatory approval, it keeps on playing a role, since it influences the BCS classification, and that is the basis for biowaivers, and also reduces the regulatory burden during the scale up and post approval changes. Now, to help in this, because there is sometimes in the early stages the availability of the compound is not there. You don't have enough compound to do a conventional solubility study. So there are number of protocols that have been developed in the industry, and they are classified into high throughput, medium throughput and low throughput protocols. So in the initial stages, what is suggested is to do a high throughput protocol, which typically starts with a DMSO based experiment. What you get in that is the kinetic solubility, and the target solubility is about 600 micromolars, and these are the buffers that are suggested. Moving over to the medium throughput studies, the sample source would be a powder. These are the buffers, and this is the target solubility. And then later you do the low throughput, which is the conventional way of doing the solubility studies, where you put it on a shaker water bath, do the sampling after 24 hours and 48 hours, and this gives you the thermodynamic solubility. The earlier two, especially the first one, will give you the kinetic solubility, and you can correlate it to the stage of the development. Say, for example, high throughput is done right at the stage of hit to lead identification, whereas medium throughput can be done when you are going to the selection of the candidate, which will go into the clinical development. And before you enter into the clinical trials, low throughput thermodynamic solubility must be known. Otherwise, you are likely to get surprises which are of unpleasant nature. Now, this is the fundamental. I won't spend too much time. There are two factors that contribute to the solubility of a drug. One is called as the crystal lattice energy, which means how difficult it is to remove the molecule from the crystal lattice. How much energy does it require? The stronger the intermolecular interactions in the lattice, more insoluble would be the compound. And the other one is called as the solvation potential, which is broadly connected to the lipophilicity of the molecule and can be quantified using log P as the parameter. They don't work in isolation, they work together. So you have to analyze both of them together to understand what is causing the problem in the solubility, and based on that, the compounds have been classified into brick dust and grease ball compounds. Both of them are insoluble, but for different reasons. Why it is important to know? The formulation intervention will change if you know the reason for poor solubility. If it is because of the solvation energy, the technologies would be different. If it is because of the crystal lattice energy, the technologies would be different. That's why it is important to know this. Now, jumping to the technologies that are at our disposal to enhance the solubility, we fortunately have a very large basket of interventions. First one are the chemical, where you can change the chemical structure, obviously without compromising the pharmacophore, the ionization state and sulfur, and the products. These need to be done right in the beginning of the discovery stage, because if they are done later on, you are going to repeat a lot of studies. And then there are physical modifications that do not change the chemical structure, and it involves polymorphic form, amorphous form, co-crystals, eutectic systems, micronization, and now nanonization. And then there are formulation approaches, which can include use of co-solvent, inclusion complex using cyclodextrins, micellar solubilization, lipidic systems, broadly the lipid-based systems, amorphous solid dispersion, and deep eutectic system. Now these are you can say the tools available in front of a pharmaceutical development scientist to improve the solubility. Now, obviously, the next question would be, how do you pick up the right tool? Is it that you can arbitrarily pick up any tool? The answer is no. You need to have a scientific basis for the selection of the solubilization tool. And to help in that, there are again lot of decision trees, a lot of guidance maps that are available. I'm going to pick only two of such examples, which are widely utilized in the industry. The first one is a very rather simple way of picking up the solubilization tool, and it was suggested by an Indian scientist, Dr. Ravi Shankar, working with Pfizer in one of the presentations he made in a conference, and that's the reference. And it considers very basic parameters of log P, melting point, and dose of the drug to choose the technology. And the technologies according to the industry could either be nanocrystals, liquid filled capsules, which are the lipid based systems, and then the amorphous solid dispersions. And you can see within the chart, one stands for nanocrystals, like a compound, if it has a log P of 2 and a melting point of 250, nanocrystals are suitable. Similarly, if you pick up a compound with a log P of 6 and a melting point of say 75, both liquid filled systems and amorphous solid dispersion could work. So that's a broad way of picking up the technologies. It does not mean that it's a watertight compartment. You will see outliers in all the cases. Some of the molecules may be amenable to a number of techniques together. So you have the choice of what you want to work with. So this is one of the simplest way of looking at it. One of more structured way that was proposed in one study that was done by European Union is the one based on developability classification system. We all are aware of BCS. So there is a developability classification system which actually emerged from BCS with few modifications. So instead of only class 2, which is high permeability and poor solubility, the drugs are now classified into 2A and 2B. And 2A are the candidates which have dissolution rate limited oral bioavailability. Solubility per se is not the problem, but the dissolution rate is the problem, whereas 2B are solubility limited. So this will give us an idea that if it is a 2A, micronization, nanonization is a good choice, but if it is 2B, then you need to go to technologies that actually improve the apparent solubility of the compound. Only dissolution rate is not going to be helpful. And based on this, then they created certain decision trees which look rather complex, but are not very complex to understand. And they suggested that you calculate two parameters, dose number and solubility limited absorbable dose, and these were meant to stratify the class 2 compounds into 2A and 2B. And based on this, the decision tree starts from this point where you have already exploited or investigated salt formation and polymorph's role in solubility enhancement. If that does not help, then you come to this stage where you see whether the D0 is greater than 1, and this is the formula for D0. If it is no, which means D0 is less than 1, then you look at the permeability. Is it the dose greater than SLAD? And if the answer is no, then you can go for a conventional type of formulation, which means neither the solubility nor the permeability is an issue. But if permeability is an issue, then you have to consider permeation enhancer approach. One point of caution here, formulation per se is not very efficient in improving the permeability, for which you usually need a chemical intervention. There are some compounds, especially the peptides that have come into the market recently, but those are few exceptions. Otherwise, permeability per se is very difficult to be addressed with the formulation approaches alone. But if the answer to this question is yes, which means the D0 is greater than 1, then you look at the permeability, and if the answer to this is no, then you go for co-milling or nanomilling, which will only do the dissolution enhancement. But if the answer to this is yes, this means you are dealing with a compound which requires improvement in apparent solubility as well. And then you have to go to different decision tree that they have suggested for solid dispersions, nanocrystals, and lipid-based systems. We'll first pick up the amorphous solid dispersions. Excuse me. One reason why we'll spend a little more time on ASDs is that it is a versatile technology, can address a large number of molecules vis-a-vis the solubility problem. And the second reason is that we have done a lot of work in this area, so I need to showcase some of those studies as well. So the amorphous solid dispersions traditionally have been classified into 4 generations, and it's basically a molecular level dispersion of a polymeric drug with the amorphous form of the drug. Amorphous form, as you know, has a higher free energy, which translates to higher aqueous solubility. So this can be exploited for improving the water solubility of a compound. And then comes the decision tree for the amorphous solid dispersion, which starts with an interesting property called as GFA, which is glass forming ability of the compound. It's a simple experiment sorry, wherein you do a heat cool heat cycle in the DSC itself, and from that you can actually experimentally find the glass forming ability class. Compounds have been divided into 3 classes: GFE 1, 2 and 3. So is the GFA available? If it is not available, you do the experiments to find it out, so it becomes available. So whether you see class 2 and 3 compounds are the ones which are good candidates for amorphous solid dispersion, because they have a relatively stable amorphous form. But if it is not class 2 and 3, which means it is class 1, then you look whether it can give twofold enhancement in the supersaturation in a in vitro assay. If it can give, if it doesn't give you, which means ASD will not work for this compound. So look for lipid system or nanosuspension, whether they can rescue this molecule or not. But if you do get twofold supersaturation and the Tm is high, Tm means the melting point, and it has ionizable groups, then you can go for micro precipitated bulk powder. If it does not comply to this, then you can think of mesoporous systems like mesoporous silica based amorphous solid dispersions. But if it belongs to 2 and 3, so it becomes a good candidate for amorphous solid dispersion. Look at the melting point. If it is not very high and if it is heat stable, you use hot melt extrusion as the technology for ASD. But if the melting point is high, you can use micro precipitated bulk powder as one of the option. And if it is heat unstable, then you look for the solubility in volatile solvents. And if it is having some solubility, you use spray drying, because these are the 3 techniques that are used for amorphous solid dispersion: spray drying, hot melt extrusion and co-precipitation. The previous 2, that is spray drying and HME, are the most exploited. Third one is not that commonly exploited. So if it is not soluble, with then again means the ASD will not be possible, and you can try out a coamorphous system by ball milling, but none of the coamorphous system till date has reached into the market. So it is largely a research curiosity as of now. Now I'm just going to give you a snapshot of the products that have been marketed using ASD. So these are commercialized products, and if you look at the numbers, the numbers have been increasing continuously. More and more products are based on amorphous solid dispersion that are coming into the market. One common example is itraconazole capsule, which comes in the name of Sporanox. Similarly, there are some other molecules which are there in the market based on the amorphous solid dispersion technology. And there are various polymers that are available to create the amorphous solid dispersion. So this is an analysis from all the commercialized products of ASD, like what polymer has been used in creating that. PVPVA takes the first prize with 33.3% compounds, ASDs that were made using PVPA, followed by HPMC-AH, HPMC, PVP, and then there are a few others which have very specific uses. If you look at the melting point of the compounds that have been marketed using ASDs, there is a wide spread. starting from 100 degrees Celsius, which is the largest category, to more than 261 degrees Celsius, which obviously has very less. So these are the molecular melting points that could be attempted with the help of ASDs. Similarly, in terms of the log P, you again have a very widespread starting from less than 1 to up to 10 value of log P, which is highly lipophilic. And the method of preparation, if we look in the commercialized product, spray drying has given the maximum number of product, followed by hot melt extrusion, and the others, as I told you, are very rarely and in very specific conditions used for commercialization. If you look at the polymer in a particular technology, that is HME or spray drying. So spray drying is given by blue color, HME by red color. So PVPVA is largely used in the case of hot melt extrusion, while HPMC-AS is more popular when you are doing a spray drying paste process. Similarly, HPMC has also been used when you are doing a spray drying process. So this is because of certain characteristics of the polymers which make them suitable for one manufacturing technique or the other. Now coming to some interesting work that we did from our lab in the area of amorphous solid dispersion. We actually generated one amorphous solid dispersion for celecoxib, and we filled that powder into the capsules. It was based on, it had three components: celecoxib, PVP and meglumine. And what came out at the end of the dissolution was this hard plug, which means instead of getting a better dissolution, in fact it was giving even a poorer dissolution, which means there was something wrong in the way the downstream processing of the amorphous solid dispersion was done. And we overcome it with the help of a multi-layered surface stabilized ASD particles using a common equipment called as the fluidized bed processor, wherein we start with certain core particles that are commercially available, and then layer by layer deposit first the ASD on the particles, followed by a stabilizing polymer, and with the help of that the dissolution could be hugely enhanced. The amorphous solid dispersion was the same, but just the way you formulated it changed the dissolution profile drastically. Another technique that we developed about a year ago was in situ ASD using freeze drying, which could be a very rapid way of investigating the strength of ASD in discovery molecule, because it is a material sparing approach and it was based on the dissolution of the drug and the polymer in tertiary butyl alcohol, because that's one of the acceptable non-aqueous solvents for lyophilization. You fill it into the opened capsule bodies, freeze dry it, you get a ASD formulation, and if you compare it to the ASD formulation that was made by spray drying, so this is the spray drying dissolution rate and this is what you get with the freeze drying. The composition was same. The reason, because the freeze drying gives you a highly porous structure, so the area available for the dissolution significantly enhances and you get a much better response. This is related to the previous one only, just to demonstrate that the physical stability was maintained, and this was the lyophilization cycle that was used for this purpose. So you can even formulate one capsule to see whether the compound has certain better oral exposure with the help of the amorphous solid dispersion. There are certain ways in which the ASD research is progressing these days, which have a very huge impact on the future. And one area that is inviting a vast interest: how can we increase the drug loading in the amorphous solid dispersion, which is right now restricted relatively to a small range of 20 to 30% of the drug in the ASD. Now that increases the pill burden, and there are attempts: can we load 50% drug, can we load 70% drug, can we go up to 80% loading in the ASD, because that will expand the application area of the amorphous solid dispersions. And we know that both the drug means its inherent properties, the polymer and the formulation play a role in increasing the drug loading. And some of the newer areas of research are based on providing a surface nano coating on top of the ASD particles, using a combination of polymers, using polymeric salts to further use the concept of microenvironment pH coupled with the amorphous form, in situ thermal cross-linking, use of surfactants, mesoporous silica. So these are some of the formulation factors that, in collaboration with the polymer type and the drug drive, are driving this innovation to increase the drug loading in amorphous solid dispersions. Another very interesting concept that has emerged is not the designing of the amorphous solid dispersion, but the fact what happens to the amorphous solid dispersion when it goes into the dissolution process. And there are very interesting findings that are coming up from a couple of labs and couple of pharmaceutical companies, especially Merck and Pfizer are doing a lot of work in this area, that what are the species that are formed after the dissolution of the amorphous solid dispersion. And they have noticed that it's a very vast array of micro and nano species that are formed post the dissolution of the amorphous solid dispersion, and they have an impact on the biopharmaceutical behavior subsequently. And one phenomena that has been now widely reported is that, suppose this is the crystalline solubility and this is the amorphous solubility. Amorphous solubility is the maximum solubility that you can achieve with the amorphous form. In between there are certain stages. So if a molecule can bypass these stages and achieve amorphous solubility, so as soon as the solubility goes above amorphous solubility, it goes into a phenomena called as liquid-liquid phase separation, where there is a drug-rich phase and then there is a dissolution medium-rich phase. Now this drug-rich phase, which could be nanospecies, starts acting as a depot from where the drug will be slowly released into the dissolution medium, and also help you in delaying the absorption process. So the intervention was not in the designing of the ASD per se, but the intervention is coming when it is dissolving in the dissolution medium. And you can see this is a very classical picture which we see a lot of places, that this is the crystalline drug solubility, and when you use a amorphous state, you actually first get a peak and then it falls quickly. So this is called as a spring effect. It goes up and then comes down. So the formulation into amorphous solid dispersion achieves this, which is called as a spring parachute effect, that after you achieve this peak, this is maintained over a biopharmaceutically relevant time frame, so that more and more drug can be absorbed. Now with the help of this LLPS phenomena, what the next generation of ASDs are proposed to achieve is called as the spring plateau effect, which means once they achieve the peak, it will be maintained like this throughout the biopharmaceutically relevant time frame. We are not talking of a very hugely lengthy time frame. A biopharmaceutically relevant time frame is 199 minutes, which is slightly more than 3 hours. So if you can maintain the supersaturation for these 3 hours, you are likely to see a huge improvement in the oral bioavailability if the drug had a solubility limited oral bioavailability. So that's the future, and these are the species which have been identified when the drug dissolves. So there could be a molecularly dissolved drug, there could be nanospecies, and if the supersaturation exceeds amorphous solubility, you go into a phenomena called as LLPS. And then there are micellar species, because there are bile salts present in the biorelevant environment. So all this array of species are present after the amorphous solid dispersion. And you can in fact correlate these to some of the delivery systems which are designed, say the micellar particles. So you, even though you started with the amorphous solid dispersion, the functional component later on is emerging as a colloidal size micellar particle. Now we come to the next category of technology which has been used to a great extent. Important thing to learn here is that amorphous solid dispersions give you an improvement in the apparent solubility. So whenever there is an improvement in apparent solubility, automatically there will be an improvement in the dissolution rate. But here is one technology which gives you improvement only in the dissolution rate. It increases the apparent solubility only marginally, which can be neglected, but it gives you a very high improvement in the dissolution rate. Now there is a decision tree even for that that is the solubility dissolution limited. So if the answer to this is yes, then you look at the melting point. Is it greater than 130? If it is not, then you either look at whether a pharmaceutical salt can be formed or not. If the melting point is higher and it has a basic pK, and then there are two conditions. You can either create a nano formulation per se or a nano formulation with precipitation inhibitor. So this is nothing but breaking down or milling a larger particle into smaller particles which are below the size of 1 micron, which means you nanonize in the range of nanometers. So the concept is very simple. Earlier there was micronization, now there is nanonization, which is nothing but creating particles whose size is in the range of nanometers. And there is no other excipient added like a polymer or anything. You only use a very small percentage of a surface stabilizer, because these small particles will have a tendency to agglomerate because of relatively high surface free energy. So to prevent that, you add a very limited quantity of either a polymer or a surfactant or a combination of both to stabilize the particles. This is important that if the basic drug, it's a weakly basic drug with a pK value of greater than 6, then there is a possibility of in vivo precipitation. So in that case, you need to include a precipitation inhibitor. But if it is not in this category, you can straight away go for a nano formulation, which here means nanocrystals, not the polymeric nanoparticles, not the lipidic nanoparticles. It simply means pure API nanocrystals. Now these are the products that have come into the market starting from 2000, which had sirolimus, and one of the most important case has been fenofibrate, where the companies have been repeatedly reducing the dose of fenofibrate using such nano-based interventions. I think the product was introduced 3 times by the innovator company by improving the formulation technology. And then you have some other compounds like naproxen. So all of them have come through the nanocrystal technology. Another interesting area where this technology is playing a role is in the long-acting injectables. So there is paloperidone palmuate long-acting injection, which is also based on the nanocrystal technology. Now, these are the 2 most established technologies for the formation of the nanocrystals, and they are called as top-down techniques, where you simply mill the larger particles into smaller particles using 2 commonly available equipment called as media milling or high pressure homogenization. And then there is a bottom-up technique where you start with a molecular solution, allow controlled crystallization to take place and arrest the particle size in the nanometer range. And this can either be done by anti-solvent precipitation, supercritical fluid technology, or spray drying. Both of them, in fact, these are the 2 most common ones that have been used industrially to bring out the products that I showed on the last slide. Now, in terms of contribution to this field, we have developed a technology called as nano-crisp technology. Now, in all the previous cases that I showed you, the product that comes out, in fact, is a nano suspension. It's not a dry powder, because this milling or this particular bottom-up approach is done in a liquid environment or a liquid solvent. So the product that you get is a liquid suspension, and that has to be then converted into a solid product. So there is a extensive downstream processing involved to convert the liquid nanosuspension into the final oral solid product. So in contrast to that, what we developed straight away gives us a solid powder, and that is the reason we called it nano crisp. Nano stands for nanometer size, CRY stands for crystalline, and SP stands for solid powder. Because you don't get a liquid product here, you straightway get a solid product. And the idea how it works is the entire events happen within the spray dryer. And please remember, in the spray dryer, the stay duration is less than a second. So within that period itself, all the events take place, and you end up with a product which contains nanocrystals. And this is the way it goes about. So you develop a clear solution of drug excipient in a solvent or a mixture of solvent, and during the spray drying, the droplets are formed, they start evaporating. Obviously there is a supersaturation, there is a complete solvent removal, crystallization of the drug and the excipient take place. The excipient is chosen in a manner that it encourages the crystallization of the drug. So we call it the crystallization inducing excipient. And then there are a formation of drug nanocrystals that are dispersed in the excipients matrix. Say for example, most common excipient that we use is mannitol. And if you look at this, so this is the one particle that you get after the spray drying. So this one particle could be of 0.5 to 20 microns. So there will be mannitol and the drug present within this particle, but the drug particles within this matrix would only be 10 to 1000 nanometers. So these nanocrystals are in fact embedded in the matrix of the mannitol, and they are not present individually separate from the excipient. So these green dots are the drug particles, whereas the purple ones are the excipient particles. Now we have 3 granted patents on these technologies in US, Europe, and India. Now this is one case study using the same technology where we work a lot with celecoxib. We have done most of our study with the celecoxib. No particular reason, we got this in large quantity in our lab in the initial stages. So that has been the molecule of our investigations in lot of studies. There's a BCS class 2 drug, shows a dissolution rate limited absorption. So obviously the nanocrystals becomes the preferred technology, because dissolution rate is the problem, not the solubility. And the excipient was mannitol, and there was sodium lauryl sulfate used in 2.5% concentration, and this concentration was well below the inactive ingredient guide recommended concentration in the US FDA's website. So these are the spray drying parameters that were used. So the inlet temperature was 120 degrees Celsius, and these are the other parameters. And this is the same picture of the particles that we got. So you can see the particles are relatively bigger in size. They are not in the nanometer size, so to say. But if you look inside one of these particles using transmission electron microscopy, then you start seeing the nanocrystals of celecoxib that are embedded in the matrix of the mannitol. And the size was calculated using a variety of techniques, using Scherer equation, which is a PXRD based methodology, and then also by dissolving the particles in a solvent and then using the zeta sizer. So the D90 was found to be around 574 nanometers with a PDI of 1, which indicates that the distribution of the size is pretty large. It's not a narrow range that you achieve, but it's okay because this is an oral product. This is not going to be used for intravenous or parenteral use. And these are the dissolution advantages that you get from this product. In a medium using 11.7, which is not a biorelevant pH, but the pK value of celecoxib is about 12. So we used this as a discriminatory medium, and you can see this is the marketed formulation Celebrex. So this was the dissolution profile, while with the NCSD we got a very high dissolution profile. And then in the biorelevant FASIF medium, this was the profile of Celebrex, whereas you can see the NCSD gives a very high dissolution rate. Now coming to the third technology, which is the lipid based formulation, and these includes self-emulsifying drug delivery system, self-nano-emulsifying drug delivery system, self-micro-emulsifying drug delivery system, and they are usually present in finally a liquid filled hard gelatin format. They can also be filled into soft gelatin, but what you usually get is a liquid filled hard gelatin capsule. So there is again a decision tree to look whether the molecule fits into the criteria of these lipid based formulations or not is by looking at the log P of the value. If the log P is low, then you can go for conventional formulation approaches or particle size reduction. But if the log P is high, then you look at the melting point. If the melting point is low, then you look at the solubility in glyceride, which is one of the lipid vehicles that is used for designing of the lipid system. So if you get a high solubility in this, you then design lipid based formulation, which can either be a SEDS or SMEDS. But if the solubility is low in this, then you look at PEG and PG solubility and their derivatives. And if it is high in that, then you develop the class 3B or class 4 of the lipid based formulation. But if the solubility is not there in either the glyceride or in the PEG and PG, then you need to go back to the other technologies for improving the solubility. Now in the case of lipid based systems, the API is actually in the dissolved form in the lipidic vehicle and other ingredient, which means the contribution of the crystal lattice energy towards solubilization is gone now, because it is already in a solubilized form. So that barrier is already gone. So the release of the drug from the droplet of the lipid into a aqueous environment would now be more facile. It would be more easier because the constraint given by the crystal lattice energy have disappeared, because we have already dissolved it in a solvent system. And they have a very complex pathway through which they are absorbed into the body, and it is also guided by the fact whether you have the medium chain fatty acid after the lipolysis phenomena or the low chain, or these are monoglyceride. These are the monoglycerides with the fraction which has C12 is less than 12. Here the C12 is more than 12, and then these are absorbed through the chylomicrons into the lymphatic system, while these go through the traditional pathway into the blood circulation. These are the products which have come into the market using the lipidic based system. So here also you see the historically right from 1941 you have products coming into the market and going up to 2021. There are some other products that have come into the market which may not be listed because of the lack of the information, and one of the most interesting cases was neo-oral or cyclosporin A, which was formulated using a lipid based system. Now, as is happening in all the areas, there are inroads being made by artificial intelligence and machine learning, which actually means replacing the human intelligence with artificial intelligence. How far that will happen, how effectively that will happen, only time will tell. But there are some significant efforts that are being made in this area. We do not work in this area, but just to make the picture complete, or to create the complete perspective, I included this part. And the idea is to identify certain molecular descriptors that affect the solubility, use them to train the models, and then try to predict the solubility of the compound. A major hurdle that is faced here is that till date no software is able to predict the melting point of the compound, that being a property which is largely unpredictable till now. So the contribution of the melting point toward the solubility is likely to be underrepresented in the outcomes of these studies. But nonetheless, these are used in the early stages of drug discovery, where we are actually not interested in the absolute values. We are only interested in rank ordering of the molecules with respect to their solubility, because we are using it as a exclusion criteria, that these are the compounds which are not worth going forward. It does not mean that the ones that have been left will all become successful drugs, but at least you have stratified a large set of library into compounds which are having druggability versus which are not having druggability. So the absolute values are not important here. They will later on in any case come when you do the medium throughput and low throughput solubility screening. So they serve the purpose in the initial stages of drug discovery very well, and hence there are a lot of efforts that are being made through these platforms. So this is one picture that I have picked up from this publication, where you can see some of the key input parameters that are used are molecular weight, log P, hydrogen bond donors, hydrogen bond acceptors, then structural, topological, electronic and environmental factor, which actually impact the solubility. So using that you create certain models and which can predict the solubility. Some of the pharmaceutical companies have integrated this successfully into their programs. So what they do is that the initial pre-formulation can be done in silico itself. You don't have to do any experiment, because you may not have the compound in enough quantity available to you. So when we look at the pre-formulation studies, they are also classified into three categories. So one would be the in silico pre-formulation studies, which are done in the computer, and then you have the high throughput pre-formulation studies, and then the third one is the conventional pre-formulation studies. And for the same compound during different stages of development, you may use different pre-formulation tools. It's not that you do the pre-formulation only once, because to do that conventional, you may not have the enough compound in the beginning of the drug discovery cycle. So the in silico predictions which then come up for certain interesting or certain molecules which show promise can then be confirmed with the experimental protocol, so that you know how far is the prediction from the actual experimentation. Because as I told you, in this the possibility of overestimation of the solubility is high, because the melting point cannot be predicted accurately. So this is the way they have been going around. So the challenges are the data quality and availability. The model interpretability is still an issue. I was taught by one of my students that it's called as a black box, because you only know what goes in and goes out. How the output came is not clear. In comparison to that, there is something called as a white box. And then obviously there are regulatory considerations of integrating AI into the pharmaceutical development. Another interesting development that is taking place over some of the pharmaceutical companies is that we know predictions can go wrong. So what they are focusing on is, can we automize the initial high throughput solubility screening to an extent that it does not become a bottleneck in the total developmental cycle. So companies like GSK have invested heavily into robotic platforms, where they are able to a very large number of compounds in a very small period of time for the determination of kinetic solubility. So they can do it in a multi-well plate. You don't need to have a analytical method to quantify, because they have nephelometric or turbidimetric methods that can give you the experimental value of solubility. So the choice whether you want to go with prediction or with a very high throughput experimental platform lies with the company, and different companies are in fact working on both the methodologies so that they can understand the comparison of the two. Now with this I reach to the conclusion of my talk. So the first conclusion is that you need to do a very careful pre-formulation profiling of the molecule that is coming from the discovery pipeline. It does not mean that you have to do the conventional pre-formulation right in the beginning. You can first do a in silico pre-formulation, followed by a high throughput pre-formulation, followed by a low throughput or the conventional pre-formulation, because only from that pre-formulation will come the information that will help you in identifying the cause of poor solubility and the technology to overcome it. Because if you recollect all the decision trees that we looked at, they wanted as an input certain very fundamental pre-formulation properties like log P, melting point and others. So you need to do a very robust pre-formulation exercise right in the beginning. And the choice of the solubility enabling technique should be guided by the physicochemical property of the molecule. It cannot be a trial and error or an arbitrary approach. It has to be a science based or science driven selection of the technology or the enabling technologies. ASDs provide a very robust platform, not because we are working into it, but there are a lot of products that are coming into market using the ASD. They are now being looked more respectfully. Earlier they were looked at with a lot of mistrust and a lot of fear, because it used to be said that amorphous form is a frustrated form because it ultimately converts to the crystalline form. But now people are developing ways and means to keep the amorphous form in the amorphous form for a reasonably longer period of time, so that its benefit could be exploited and the shortcomings could be hidden by technology platforms. There are innovative materials and techniques that are driving the advancement of these new age ASDs, and as I told, dissolution, understanding of the dissolution phenomena is going to be the next in thing for next 5 to 10 years, that will drive the designing of the amorphous solid dispersion. Nanocrystals, although a good commercial platform, offer advantage only in terms of the dissolution rate, not in terms of the apparent solubility. And lipid based formulation can provide an interesting additional advantage of permeability apart from the solubility. And AIL ML are providing exciting opportunities in technical solubility related issue. I hope they don't become as exciting as to replace the human intelligence, because that would be difficult for the pharmaceutical formulation fraternity, because they will be largely replaced by machines and not be able to get proper jobs. With this, I just give you a glimpse on a lighter side of the NIPER campus, where I've been there for last 25 years or so. So this is the diversity that is available in the NIPER. So apart from the human diversity, we also have diversity in terms of the flora and fauna, and this is actually clicked in the campus of NIPER by one of our own student, and this was the group who have contributed largely to the work that I have shown, although this is the last year's group, but some of the students are here, especially the PhD students. So with this, I thank you all for your attention. So thank you, Prof. Hello. So thank you, Professor Bansal, for outstanding insightful talk. So,
Hello. Okay. So thank you, Professor Bansal, for outstanding and insightful talk. So due to the time, we can take one or two questions. And I want to mention that there is an interaction session 101, interaction session with scientists at 2:30 p.m. and with students also at 3:30 p.m. So most of the question can be discussed there. So now we can go for one or two questions if it is there.
Thank you for wonderful talk, sir. Actually, I was wondering, what about the scalability of these techniques? Because these are theoretically they look very promising, very forward-looking, but when it comes to the scalability, where the industrial skills are required, the techniques like this vaporization and others, cryo-related techniques, would it not be very expensive and difficult to scale?
So are you referring to some specific technique that I referred to?
So this was freeze drying, like you showed something in the capsule and freeze drying techniques, and you optimize there. And when it comes to the scalability, like you translate it from the lab scale to the
So in fact, it's not the first time that freeze drying has been used for the production of oral solid dosage forms. There is a very well established platform called as Zydis technology, where oral soluble tablets, they are actually not tablets because they are not compressed, they are produced using freeze drying as a technology. Obviously there are certain modifications to the existing lyophilization equipment that would be required, but if you have a line related to the Zydis technology, that can be integrated for technologies like this. Why I specifically showed this slide is that in a setup where the material is less, a material sparing approach could be something like this, because if somebody has worked with spray drying, they know the yields in the lab scale spray drying, Amit is agreeing with me, will not go above 30 or 40%. 60. That would be after 3 years of optimization, I believe. So similarly, HME requires a lot of material. There are some new technologies that are coming up. There's a technology called melt prep, but it's not readily available. So as a material sparing approach in the initial stages of investigation, to see whether ASDs will work or not, this can be utilized, but it's a far way to go in terms of the commercial technology. And the rest of the techniques that I showed, they are all commercially feasible, because I showed you a lot of products that have been commercialized using them.
Thank you, Professor Bansal. I have just one query, that at very beginning of synthesis, when we synthesize compounds and submit for biological assays, so what would be the ideal form, whether it is amorphous or crystalline? What should we consider to be the first sample?
In fact, that becomes a non-question in the sense that all the biological assays are done in the dissolved form. You will typically dissolve it in DMSO. So if you do a DMSO based assay, whether you start with amorphous or crystalline or anhydrous, actually doesn't make any difference, because it's a solely solid state property, but once it goes into the solution, it has no further meaning.
Okay, so both works out. If we
both work, because you are at that time looking at the molecular properties, how the molecule is interacting with the receptor.
Okay, so if we look at the developability probabilities at the later stages, at the very beginning, so we should start with amorphous forms, like can we go to amorphous ADCs and all?
In fact, in the initial stages, mostly we are not even aware what form we are dealing with, because as the studies are progressing, your chemical synthesis is also scaling up from milligram to say gram. So usually what is the best practice is to keep recording a PXRD and a DSC, just to keep a record of what went into which trial, because at that time you have still not done a complete solid form screening. But at least retrospectively, if you have to go and analyze that what happened or what was the difference between the two batches, you have that data available with you. Now to answer your question, in most of the early stages, the chemists give amorphous compound to the biologist or to the formulation scientist, because their crystallization processes have not been optimized till that point. And similarly, the solubility, that also is done in the high throughput. in fact gives you the kinetic solubility, which is more representative of the amorphous form, and hence would always be higher than what you will get in the low throughput solubility studies later on. So that has to be factored into your calculations.
Okay, thank you.
Thank you so much, Professor Bansal. The remaining questions can be discussed at 2:30 at the hall number 2, and also students are encouraged to join at 3:30 for the discussion. So now may I request all the dignitaries to the dais again, please. So dignitaries to the dais, please. Sir. So I am delighted to introduce our chief guest, Professor Prabodh Trivedi, Director, CSIR-CIMAP, Lucknow. He did his BSc and MSc from Lucknow University, and then PhD from RML Awadh University, and did his postdoc from University of Maryland, College Park, USA. He joined National Botanical Research Institute, that is CSIR-NBRI, in 1994, and he worked there as senior principal scientist up to 2020, and then he joined as director CSIR-CIMAP, that is Central Institute of Medicinal and Aromatic Plants, in 2020. His contributions are recognized globally. He has been awarded with several prestigious awards, and he is on editorial boards of various scientific journals. I am going to mention some of them. He is recipient of J.C. Bose National Fellowship. Sir, little bit, sir. Sir, please, a lot of students are there, so let them know, sir, please. It's okay. So he's also fellow of FNA, FNASC, and also the member of advisory committee of biotechnology UPCST. So, and also sir published more than 180 papers, book chapters, H-index more than 56, and he is well known in the field of plant research in India, and we are privileged to have sir to us here today with us. So please, thank you so much.
Dr. Radha, Director CDRI, Professor Bansal, Dr. Yadav, Dr. Gayan, and all the colleagues from CSIR-CDRI and students. I feel privileged to be part of this series basically, which is innovation in health. Though I'm not from this area, however, I really loved today's talk. Starting from basics, it went on to the most advanced things, that how the solubility can be changed or increased. As I see CSIR-CDRI, it has been working in this area since inspection, and number of drugs have come into the market, scientifically validated drugs basically, and CSIR is proud of the contribution of CSIR-CDRI basically. And I'm really happy that in some of the inventions, CSIR-CIMAP has been part. If you see anti-malarial drug artemisinin, CSIR-CIMAP developed varieties like Sim Sanjeevani and Sim Arogya, as well as process technology, that how molecule can be isolated from those plants, and after that went to the market. And very recently, research on Videnia for anti-stroke as well as bone health, CIMAP is collaborating with CDRI. So two institutes in one city collaborating with each other, I think that can make difference. With number of things can come because in several cases medicinal plants play a very important role. Today's talk by Dr. Bansal gave an in-depth information about solubility of different molecules, and how what are the challenges and how we can overcome those things. Dr. Bansal pointed out several things like absorption, distribution, metabolism, excretion, like everything is affected by this solubility issues. And what he said during his presentation, like fail early, fail cheaply, and that should be the thumb rule. Like we keep on working for years without looking at those things, I think. So thank you very much for bringing this point here, like fail early and fail cheaply. And as I see that solubility is in not only this case on drug development, but it plays major role in different aspects, be it the interpersonal relationships and other things. Like solubility is the main case. If you are really soluble, you can make difference in all every part. As I was listening the presentation, he talked about like chemical modification, for different formulation techniques, advanced techniques like co-crystals, microemulsions, nanoemulsions, everything. Like and those were really, I think, some of the students sitting here, those who came from different parts of the city, or those who are joined online will be beneficial about those things. What I feel that most of the thing like ASD or log P or melting point you talked about during this presentation, what I felt that those are basically for single molecules. And because most of the time we work on phytopharmaceuticals, then what about there are 10 molecules in one, and we don't know what is the log P for each molecule and melting point, then how to go about it? That is a big challenge. I think if you have your views on this, maybe during the discussion at 2:30, and after that you can just because most of the scientists here, I guess, working on phytopharmaceutical pipeline, and where single molecule is really difficult to work on, so that will be like a good thing to study. And as I see that though there were solubility issues, but number of drugs, like you have already shown, that have come. And if you see plant-based like Taxol, which has very less solubility, 0.3 microgram, I think, per ml. And so number of techniques have been adapted, and after that that came to the market. Similarly, like nowadays everybody talk about curcumin, which has very low solubility, low bioavailability, but like nanocarriers and liposomes have been worked out, and which have made it biosoluble basically. So we need to take a number of different approaches, multidisciplinary approaches, to make things soluble and can come out with drug development. So thank you very much for this was a learning talk for me. Thank you very much, and I really thank Radha, Gayan, and Prem for giving me opportunity to be here to learn new things. Thank you very much.
Thank you so much, sir. So now it's the time to felicitate our speaker. So I will request chief guest Dr. Prabodh Trivedi and our director ma'am Dr. Radha Rangarajan both to felicitate our chief speaker Professor Arvind Bansal. Thank you, sir. Thank you, ma'am. So now I'll request director ma'am to felicitate our chief guest Dr. Prabodh Trivedi, please. So in the meantime, I will request Dr. Niti Kumar, Dr. Arvind Khatri to come on the stage for a group photo. So thank you. So now I will hand over to Dr. Prem Prakash Yadav to propose vote of thanks, please.
So, good afternoon, everyone. Thank you, Dr. Jia, for seamlessly taking us through this session. As we come to the close of this event, it's a truly memorable lecture, and we all are benefited. It's also part of our 75th year celebrations. So, it's congratulations to all of us on this occasion. It's my distinct honor and privilege to propose vote of thanks. On behalf of whole CDRI, I would like to extend my heartfelt thanks to the chief guest, Dr. Prabodh Trivedi, and the esteemed speaker, Professor Arvind Kumar Bansal, for accepting our invite and making it possible for this learning experience for all of us. So, a special note of thanks and gratitude to the director CSIR-CDRI, Dr. Radha Rangarajan, whose vision and whose initiative of innovation in healthcare series lectures has let us gather over here and listen to Professor Bansal. We have witnessed 4 lectures during last year, and this is the first lecture of this series for this year. We will be having 2 more lectures coming up in this year. Thank you, ma'am, for all your support and guidance. I am also thankful to the wonderful audience, which comprises of students from Lucknow University and students from Rameswaram Institute, and I hope weather must not have refrained them from joining us in good numbers. So thanks to the team who coordinated their visit, and all the faculties and students from Lucknow University and Rameswaram Institute. I'm also thankful to the audience who have joined online. There are more than 100 participants. I have been just informed that it's more than 100 people who have joined online this session. So they must have got benefited from this lecture. Thanks to all of them. This event would not have been possible without efforts of organizing committee members, Dr. Gayan, Dr. Niti, Dr. Arvind, and I'm especially thankful to Dr. Gayan for taking all the pain to listen to this wonderful lecture. Thank you, Dr. Gayan. I'm also thankful to the student volunteers, Devalina and Shivam, for coordinating the session and managing the stage. We sincerely acknowledge the efforts from all the backstage team, auditorium team, led by Santoshji, Sumit, Arvind, IT team, audiovisual team, camera person, Kazim, who also made this beautiful flyer for this event, and PME, academic unit Sanjeev and Hijaz, who coordinated this event for student participants from outside CDRI. I'm also thankful to the canteen horticulture team for the beautiful stage management. We also acknowledge necessary help from the director's office, from administration, finance and accounts, stores and purchase, and thanks are due to Bhaskarji, Sanjeev Sekharji, and Vinayji and their whole team. I may not be able to take all the names, so I wish to extend my heartfelt thanks to everyone whosoever is involved in organizing this event, and all of you for gathering here in time and making this event a huge success. So once again, thank you all, and before we conclude this session, may I request all the dignitaries to join us in the front rows and all the audience to come closer for a family photograph. Thank you all.